Hi everyone, I'm Fengyuan Huang, a graduate student from the University of Alabama at Birmingham. The topic of my poster is Baxambra, a circular bacterial genome assembler. Accurate bacterial genome de novo assembly is fundamental to understand the evolution and pathogenesis of bacterial species. Thanks to the third generation sequencing data and published bacterial draft genome have grown dramatically. However, the structure of bacterial genome is circular and they contain many repetitive sequences that makes genome assembly very challenging. Their existing assembler apply only long reads, only short reads, or hybrid reads, while they are far from optimal. Most of the long read assemblers are not designed for bacterial genome as they are not aware of the circular structure. Since stories cannot cover the lipid fragments, it is difficult for them to solve the lipid regions. Hybrid reads assembler like unicycler iron spade plus recompile, which is more likely to create fragment assemblies and many structural errors. So considering all these reasons, we develop a new method, Baxambra, specific for bacterial genome. Baxambra accept only nanopore or PacBio long reads or hybrid reads include short and long reads. In the long read only mode, it first select the longest read that cover about 50x of the genome and collect the selected long reads by the remaining reads to produce an initial assembly. The two ends of the initial assembly undergo the assembly by the subset of reads that map to the region, and the content generated by this reads replace the two ends. After merging the initial and the end content, we generate a complete and circular assembly. Finally, Baxambra also arrange start position to the DNA gene and polish the circular assembly with the high depth long reads. In the hybrid mode, the key steps are the same, but it combines the accurate stories instead of long reads for the collection process. We first test on simulation data from bacterial strain Mycoplasma arginini and compare with the long read assembler WTBG2, Fry, and Canoe, and hybrid assembler unicycle, etc. The performance were evaluated by the Quast. All assembler accept hybrid space generate one complete contact with comparable lengths. WTBG2 unicycle hybrid mode got one misassembly style represent large scale structural error. The base accuracy were measured by the number of mismetrics and index per 100 kilobase pair. Both Baxambra and Fry have the lowest number of mismetrics, and Baxambra hybrid mode can eliminate all the mismetrics and index. So overall, Baxambra surpassed the other tools with the simulated data and construct the most accurate genome sequence. We further text on the two other Mycoplasma arginini and polyphenol strain sequence isolated from infected patients. Since these two strains diverge significantly from the published genome, we cannot use Quast to evaluate the assembly quality. So we introduced two other metrics, the number of supplementary alignment and number of supplementary cluster. They indicate that there is alignment ambiguity due to the structural error based on the fact that we did not expect to see the supplementary alignment or cluster in the error-free assemblies. The performance on amphipolyphenol shown as the bottom part. In table 2, all the assembler produced one complete contact. However, Baxambra outperformed the other assembler by generating a minimum number of supplementary alignment. The performance on Afginini show as the upper part, Baxambra, WTBG2, and Canoe got one complete contact while the other generate more than one, and Baxambra got the least number of supplementary alignment and no cluster. To further evaluate the accuracy of Baxambra, we use PCR amplification on 76 selected low complexity location and perform single sequencing. We map the amplicant sequences to the individual assembly and calculated the index and mismetrics between PCR amplicant and the context. There are most number of PCR reads could be mapped back to the Baxambra context and the minimum number of mismetrics and index in Baxamber assemblies. With additional taxon 14 bacterial PacBio sequencing data from the NCTC 3000 project, which provide assembly reference. Compared to the other tools, Baxamber achieved the least number of contact, and the M50 are very close to the reference. And this suggests that Baxamber can tend to generate complete genome, and Baxamber also have best performance in resolving genome duplication sequence and generate the least number of misassemblies. For the base accuracy, overall, unicycle have the highest number of indel and mismatches, while Fry, Canoe, and Baxamber got a lower number. So this result indicate that Baxamber are also capable to assembling PacBio sequence with less base error. So in summary, Baxamber can generate circular genome assembly that have the least misassemblies on both simulation and real sequencing data. Therefore, Baxamber provide a better solution on bacterial genome assembly using long read or hybrid reads, which is available for downstream and other bacterial genome analysis. Thank you.